Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing CUCET MSc Entrance question paper. The subject code is PGQP78 and the subject is Applied Microbiology. This was conducted in the year 2022. I will be solving only the biology answers. Okay, coming to the first question. Nitrosomonas, a nitrifying bacteria, is a chemoheterotroph, chemoautotroph, photoorganoheterotroph, photolithotroph. So, Nitrosomonas, it is involved in oxidation of ammonium to nitrite. So, it is a chemoautotroph. Answer is option 2. Which one is not the branch of microbiology? Immunology, virology, bacteriology, geology. Oh, that everybody knows. It is very simple question. Next one. Secondary metabolite is synthesized by which phase of growth? So secondary metabolite, it is usually synthesized in the stationary phase of growth. Okay, so the answer is option 4. Then next one. Spirulina, which contains about 65% protein of its dry weight, belongs to. So spirulina, it belongs to cyanobacteria. So answer is option 2. All these are direct questions. The end product of the denitrification process is. So in denitrification, nitrate is reduced to nitrogen gas. So the end product of denitrification process is nitrogen gas. Answer is option 3. Which antibiotic inhibits transcription of DNA during protein synthesis? Penicillin. Penicillin, it inhibits cell wall biosynthesis. Rifampicin, it inhibits bacterial RNA polymerase. And RNA polymerase, we all know it is responsible for transcription. So they have asked which antibiotic inhibits transcription of DNA during protein synthesis. It is rifampicin because it inhibits bacterial RNA polymerase. Chloromycetin is a translational elongation inhibitor. And next one. Which is not a structural gene of lac-operon? So lac-operon has got 3 structural genes, that is Z, Y and A. I is not a structural gene because it codes for the repressor protein in lac-operon. So answer is option 3. Next one. Ribozymes, they are catalytic RNA molecules. Are catalytically active molecules of. So it is RNA. Answer is option 3. Type 4 secretion system is involved during gene transfer in. So type 4 secretion system, it is involved in conjugation. Answer is option 1. Which non-leguminous plant forms symbiotic association with rhizobium? Casuarina, it is Frankia. And Parasponia, it is rhizobium. So answer here is option 2, rhizobium. How many ATP are produced during glucose fermentation? So during the first stage, glucose is converted into pyruvate through the process of glycolysis, and 2 ATP and 2 NADH are produced. Now what happened to this pyruvate is that it is later broken down into lactic acid or ethanol and carbon dioxide is released. ATP is not produced during this stage. And 2 NADH generated during glycolysis, it is consumed in this stage. So thus a total of only 2 ATPs are generated from the fermentation of a glucose molecule. So the answer here is option 1, 2 ATP. Next one. Which fungus is used for lactic acid fermentation in a bioreactor? So for lactic acid fermentation, you use Rhizopus oryzae. Answer is option 4. Next one. Which of the following bacterium is used as biocontrol agent? So biocontrol agents, they are natural enemies of insect and pests. So which bacterium is used as biocontrol agent means it is Pseudomonas. Next one. Which of the following is a mobile electron carrier in the mitochondrial electron transport system? So it is mobile electron carrier, it is ubiquinone. Answer is option 2. DNA replicates in which phase of the cell cycle? Everybody knows it is S phase. So coming to the next question. Agarose is extracted from, it is extracted from brown algae. So brown algae are called as seaweeds. So the answer here is option 2, that is seaweeds. Which technique is prepared in the separation of fatty acid methyl ester from a mixture? So it is the gas chromatographic method that will be used. Which pyrimidine base contains an amino group at carbon 4? So it is your cytosine that contains an amino group at carbon 4. This is the structure of cytosine. This is first nitrogen for first carbon atom or nitrogen atom first. The numbering starts from here. 1, 2, 3, 4, 5, 6. So at the fourth position you have got an amino group attached. So it is cytosine. Answer is option 1. Now next question. For the accurate mapping of genes, dash is used. So for the accurate mapping of genes, 3-point mapping is used. So the answer here is option 1. Then next one. The photosynthetic bacteria which is not an autotroph. So out of these, the rhodospirillum, it is not an autotroph. Answer is option 3. Name the amino acid which is used as a precursor for indole acetic acid for promoting plant growth. So it is tryptophan. Answer is option 2. Next one. Which forces mainly maintain the three-dimensional structure of the protein? So it is both covalent and non-covalent forces that maintain the three-dimensional structure of protein. Both covalent and non-covalent are involved. Then next one. Bacteriorhodopsin, a pigment which helps in the phototrophy and ATP synthesis, is found in. So it is found in Halobacterium. Answer is option 1. In polysaccharides, O-glycosidic bond forms between. So a O-glycosidic bond, it is formed when anomeric carbon of the sugar. So anomeric carbon of the sugar establishes a link with the oxygen atom in the hydroxyl group of the alcohol. So the answer here will be anomeric carbon and alkoxy oxygen. Answer is option 3. Next one. Penicillin is produced in which of the following processes? So penicillin is produced by fed batch fermentation. Answer is option 2. The viscosity of double-stranded DNA molecule is lost due to. It is lost due to denaturation. As a result of denaturation, the two strands separate and viscosity of DNA is reduced. Then next one. Transfer of gene of choice from bacteria to higher plants is mediated through. So binary plasmid system. You can see that in the case of agrobacterium mediated gene transfer to plant systems. There you use binary plasmid system. Then next one. Which of the following act as an infectious agent during cyanobacteria plant symbiosis? So it is Hormogonia. Answer is option 3. Then next one. In the presence of oxygen, C3 plant photosynthesis is inhibited due to. So it is inhibited due to Warburg effect. In the presence of oxygen, photosynthesis is inhibited in C3 plants. That is called as Warburg effect. Then Bt toxin is a protein coded by the Cry gene. Answer is option 2. Don't confuse it with the option 1 because there the spelling is wrong. It is given as CYR whereas in option 2 it is CRY. So the answer here will be option 2. Then next one. In normal phase chromatography, they have asked what is the mobile phase and the stationary phase. In normal phase chromatography, it is the mobile phase is non-polar and the stationary phase is polar. So the answer here is option 2. Who is the father of environmental microbiology? It is Sergei Vinogradsky. So answer here is option 2. And cystic fibrosis. Cystic fibrosis, it is an autosomal recessive disorder. Answer is option 4. All these are just direct questions. That's why I am not explaining much. Then next one. Which one is known as the topographic abiotic factor? So topographic abiotic factor out of these is earth surface. Answer is option 4. DNA is negatively charged because of the presence of phosphorus in it. Phosphate groups in it. So DNA is negatively charged. Molecular scissors used in genetic engineering. They are restriction endonucleases. Restriction endonucleases are usually called as molecular scissors. They are used to cut DNA. Carrageenan, a jelly-like substance, is derived from the marine alga known as Irish moss, or botanically it is called as Chondrus crispus. So answer is option 3. The study of the group characteristic of a population, their changes over time and prediction of future change are known as. So it is called as demography. The next one. The element necessary for translocation of sugars in plant is. So for translocation of sugar in plant, you need boron. Answer is option 2. Then next one. Ecological succession is A. So it is a dynamic process. Ecological succession, it is a dynamic process. Next one. Which biological agent acts as a mutagen? So biological agent acting as a mutagen is transposon or jumping genes. They keep moving from one place to the other. So if it is been incorporated into a particular gene, the function of the gene will be lost and it gets mutated. So biological agent act as a mutagen is transposon. Who discovered first azotobacter as nitrogen fixing organism? So Martinus Beijerinck. Answer is option 2. Which of the following cell organelle does not contain DNA? Nucleus has, chloroplast has, mitochondria has, lysosome doesn't have DNA. Answer is option 4. Given below are two statements. One is labeled as assertion A and the other is marked as reason R. So assertion A. All bacteria have prokaryotic cell type. Okay, that is correct. Bacteria do not contain any membrane enclosed organelles. That is the reason why bacteria have prokaryotic cell type. So both A and R are correct. And R is the correct explanation of A. Next one. Again assertion and reason. Assertion. Bacteria contain 16S rRNA gene which is used in biotyping. The 16S rRNA gene is present in the small subunit of ribosomes. Now both are correct, but R cannot be the explanation for A. Because some regions of this 16S rRNA gene, they render species-specific signature sequence which is used in identifying bacteria. That is the reason why 16S rRNA gene is used in biotyping. Then next one. Again assertion and reason. T4 phage infects the E. coli cell. T4 phage is capable of undergoing only a lytic cycle. Yes, that is correct. They infect the E. coli cell and they undergo lytic cycle. They break open the E. coli cell and come out and again infect the next E. coli cells. So both A and R are correct and R is the correct explanation of A. Next one, again assertion and reason question. Assertion A. The reduction of molecular nitrogen to ammonia requires high activation energy. Molecular nitrogen is an unreactive gas with a triple bond connecting the two nitrogen atoms. So that is the reason why reduction of molecular nitrogen to ammonia require high activation energy. So both A and R are correct and R is the correct explanation of A. Next one. Saccharomyces grows both under aerobic and anaerobic conditions. That's correct. Under aerobic condition, it utilizes glucose for the production of biomass, and under anaerobic conditions, it ferments glucose into ethanol and carbon dioxide. Yes, under aerobic condition, it undergoes glycolysis. Glucose undergoes glycolysis and result in the production of biomass, whereas in anaerobic condition, it result in the fermentation of glucose to carbon dioxide and ethanol. So in both A and R are correct and R is the correct explanation of A. Next one. Again assertion and reason. A unicellular microorganism divides by binary fission and forms sigmoidal growth curve during its growth. During death phase, some cells are able to survive and grow. That is correct. In the stationary phase, secondary metabolites produced by cells are utilized as nutrients for its survival during death phase. That is not correct. So A is correct whereas R is not correct. Then next one. During bacterial transformation, single-stranded DNA is transferred into cells. Endonuclease located on the cell wall cleave one of the strands of DNA. So here, during bacterial transformation, single-stranded DNA is transferred into cell. That is correct. Endonuclease is located on the cell wall. It is located on the cell membrane, not on the cell wall. So A is correct, but R is not correct. Then next one. Assertion. NADP plus is reduced to NADPH in non-cyclic photophosphorylation. That is correct. NADP is a coenzyme which is reduced to NADPH2. The hydrogen required for formation of NADPH2 comes from hydrolysis of water molecule. So both A and R are correct and R is the correct explanation of A. Then next one. Given below are two statements. One is labeled as assertion A and the other as reason R. Assertion. An important chemical reaction, nitrogen to ammonia, is involved in nitrogen cycling in an ecosystem. The organism associated with this chemical reaction is nostoc. So both A and R are correct and R is the correct explanation of A. Given below are two statements. One is labeled as assertion A and another as reason R. Assertion. Mitochondria and chloroplast have their own DNA. Everybody knows that. Now reason. They are called semi-autonomous organelles since their DNA encode proteins required for metabolic function of mitochondria and chloroplast. That is that is correct reason. They sorry. They are semi-autonomous organelles and their DNA, that is mitochondrial and chloroplast DNA, they produce proteins which are required for the metabolic functioning of mitochondria and chloroplast. So both A and R are correct and R is the correct explanation of A. Then again assertion and reason. Assertion. E. coli is a coliform bacterium. Yes, that's correct. Coliform bacteria are indicator of water quality. That is also correct. So both A and R are correct and R is the correct explanation of A. Then next one. Again assertion and reason. There are three stop codons: amber, ochre, and opal. UAG is amber. That's correct. UGA is ochre. No, UGA is not ochre. UAA is ochre and UGA is opal. So A is correct but R is not correct. Next one. Heterocyst contain nitrogen fixing enzyme nitrogenase under anaerobic condition. That's correct. Heterocyst lack pigment system 2. That is also correct. So both A and R are correct and R is the correct explanation of A. Then next one. Again assertion and reason. Assertion. Lambda phage infects K12 strain of E. coli. Yes. Lambda is a temperate phage which causes lysogeny in E. coli. Yes, that is also correct. So both A and R are correct and R is the correct explanation of A. Then again assertion and reason. Chlorophyll A is universal chlorophyll found in organisms, algae to higher plants. Yes, that's correct. Chlorophyll A forms reaction center in photosynthesis. That is the reason why chlorophyll A is universal chlorophyll. So both A and R are correct and R is the correct explanation of A. Then next one. Again assertion and reason. Assertion. Roots of higher plants secrete carbohydrate, amino acid, vitamins into rhizospheric soil. Yes. Plant growth promoting rhizobacteria convert amino acid tryptophan into IAA. Yes, that is the reason because these roots of higher plant they secrete amino acids and the rhizospheric bacteria they convert amino acid to tryptophan IAA. IAA is indole 3 acetic acid and it is an auxin. So both A and R are correct and R is the correct explanation of A. Next one, assertion. Troposphere lies above lithosphere and hydrosphere and contain aerosols. Yes. Bioaerosol in the troposphere contains spores of fungi. Yes. So both A and R are correct and R is the correct explanation of A. Then next one. Again assertion, reason. Proteins upon hydrolysis with concentrated HCl at 100 degrees Celsius for 20 hours yield amino acids in the form of their hydrochlorides. Very correct. Threonine is destroyed during acid hydrolysis. That is also correct and that is the correct reason for this particular assertion. So both A and R are correct and R is the correct explanation of A. Nucleotides may have 1, 2 or 3 phosphate groups covalently linked at the 5' OH of ribose. Correct. The hydrolysis of ATP is an exergonic reaction. That is also correct, but it is not the reason for this particular assertion. So both A and R are correct, but R is not the correct explanation of A. The next assertion. Alleles separate during gametogenesis. Correct. Separation of factors is due to segregation of chromosomes during meiosis. That is all the reason for why alleles separate during gametogenesis. So both A and R are correct and R is the correct explanation of A. Next one. As statement 1. Given below are two statements. So here it is statements and we have to choose the appropriate answer from the options given below. So statement 1. The nucleolus plays a major role in ribosome synthesis. That is correct. Hydrogenosomes have double membrane and carry out metabolic processes that generate methane gas. It is not methane gas, it is hydrogen gas. So statement 2 is not correct. So statement 1 is correct but statement 2 is incorrect. Next one. Amino acids vary in their acid-base properties and have characteristic titration curves. That is correct. Glycine has one type of titration curve, histidine has another type. So they vary in their acid-base properties and have characteristic titration curves. Some proteins contain permanently associated chemical components in addition to amino acids. These are called conjugated proteins. So that statement is also correct. So both statement 1 and 2 are correct. Next one, match the following. They have given list 1 with bacteria and list 2 with where they will be found. Usually the environment in which they will be found. So psychrophilic bacteria. Psychrophilic bacteria, they are usually found in Antarctica, cold conditions. So A2. Thermophilic bacteria are found in hot water springs. Hyperthermophiles are found in thermal vents. That is 1. And mesophilic bacteria, they are found in the normal environment. 3. So the answer here is A2, B4, C1, D3. So coming to the next question. This is list 1 and this is list 2. You have to match them. So DNA polymerase, that is involved in DNA replication. A3. Okay, you can cut off the 2 and 3. 1 and out of 1 and 4, which is correct, we have to see. A3. Cybrid. Cybrid is fusion of cytoplasm with whole cell. B4. Yeah, for first option is correct. We will see others also. Cosmid. Cosmid is plasmid vector with cosite. DNA probe. A small piece of DNA radioactive isotope labeled. That is called a DNA probe. A small piece of DNA which is labeled, which is radioactively labeled. So the answer here is option 1. Then next one. Collection of genes of an individual. That is called the genotype. A2. Law of heredity. It is Mendel. B1. So your answer comes out to be option 4. A2, B1, whereas in second option A2 and B3. So that is not correct. It is B1. Laws of heredity was proposed by Mendel. Spliceosomes, they contain are found in fungi. DNA fingerprinting, zinc finger analysis. C4 and D3. Then next one. Enrichment of water body by nutrients. That process is called as eutrophication. A2. Animals consume both plant and animals. They are called as omnivores. Thermosphere, it is a division of the atmosphere. So answer is option 4. Largest ecosystem in the world is the oceanic ecosystem. So answer here is option 3. That is A3, B1, C4 and D3. Next one. Pedigree analysis. Pedigree analysis is quantitative genetics. Then bar bodies are found in female sex chromosome. Male heterogamety. Male heterogamety is found in human. You have X and Y chromosome. Sex determination was first studied in the plant Melandrium. Answer is option 4. So the first answer A1, B2, C3, D4. Then next one. Plasmid. Plasmid is extra chromosomal DNA. A2. Cosmid. Cosmid, plasmid contains hybrid plasmid contains lambda DNA cohesivence. C4. B4. A2, B4. BAC. BAC is plasmid containing replication origin of E. coli factor. and yak is used for cloning in yeast. So the answer here is option 4, A2, B4, C1, D3. Then next one, lag phase. During lag phase, synthesis of macromolecules take place. During log phase, primary metabolites are produced. Stationary phase, secondary metabolites are produced. And during the death phase, it is cryptic growth. So the answer here is option 3, A1, B3, C2, D4. Then next one, edible fungi. Edible fungi is agaricus. A2. Penicillin is will cause Penicillium chrysogenum. Penicillin is obtained from Penicillium chrysogenum. So B3, A2, B3. Rust of wheat is caused by Puccinia triticale. C4, and budding is found in Saccharomyces. B1. So answer is option 2. Next one, microcystis. Microcystis, it produces toxin. A3. Anabena, it is used as biofertilizer. Vaucheria will form xenozoospore, and Edogonium has got an anandrous male. So answer here is option 4. A3, B1, C4 and D2. Then hormones. Auxin. Auxin, it influences root formation. A3. B. Cytokinin, they stimulate the growth of the late buds. Gibberellins, they help in stem elongation, and abscisic acid is involved in stomatal closure. So the answer here is option 3, A3, B2, C4 and D1. So that's it. Thank you for watching my channel. Please do like, share and subscribe. I have also uploaded papers from biochemistry, life science and plant biotechnology also. You can go through them also if you are interested. So thank you for watching my channel.